Hello, everyone. Welcome to the Network Physiology webinar series. My name is Alireza Mani, and I'm one of the organizers of a series of webinars on the emerging field of network physiology. This is our second session of the series, which is dedicated to network physiology in extreme environment. This session will feature 3 talks, with each talk lasting approximately 20 minutes. After the third talk, we will have a question and answer session. I would like to thank the speakers who accepted our invitation and also the Physiological Society for making this event possible. My colleague, Dr. Tope Oyelade, is the other organizer of the webinar series. Tope, would you like to say a few words?
Yeah, thank you very much, Alireza. And again, thank you all for attending this webinar. I'll just take the moment to go through some of the housekeeping housekeeping instructions specifically. I think the first thing to say is that please be aware that this webinar is being recorded for the purpose of being published in future. So we'll kindly ask you to keep your microphones and video cameras off for the duration of this meeting. Again, today's session is going to be talking about network physiology in extreme environment. In terms of, we know a lot of you are going to have some very fascinating questions, and we're really looking forward to answering them. Please submit any questions through the chat. You can submit your questions at any time, either during or after each talk. Questions can be typed at any time, like I said, and all questions would then, after the talks, be directed to the speakers and chairs during the Q&A session at the end of the talks. I think thank you very much, Ali. I think we can go ahead now. Thank you.
Thank you, Tope. Thank you very much. So excellent. So it is my pleasure to introduce our first speaker, Dr. Joe Costello. Dr. Joe Costello is the Associate Head of Research and Innovation at the School of Sport, Health, and Exercise Science at the University of Portsmouth. He is also an associate professor and the team lead of the Extreme Environment Group. Dr. Costello is an expert in extreme environmental physiology, with research focused on understanding how various stressors, such as heat, cold, and hypoxia, affect human performance. He is an active contributor to the academic community, regularly delivering invited talks at the national and international organization. He also holds a position on the editorial board of Experimental Physiology, our beloved journal of the Physiological Society, and also he serves as a methodological editor with the Cochrane collaboration, excellent for evidence-based science. Prior to joining the University of Portsmouth, he completed his PhD at the University of Limerick in Ireland, followed by 3 years of postdoctoral research at Queensland University of Technology in Australia. I have personally learned so much from Dr. Costello through our collaboration on various projects. His depth of knowledge, clarity in research, and collaborative spirit makes him an outstanding colleague. So thank you, Dr. Costello. We are delighted to have Dr. Costello here today and to share his experience and expertise on the application of network physiology in extreme environment. Dr. Costello, thank you.
Thank you very much, Alireza and Tope. Hopefully, everyone can see my presentation. Yes, excellent. Good afternoon, everybody. It's great to be here. A huge thank you to the Physiological Society for the opportunity to present on this session, and also to Alireza and Tope for the invitation as well. So my role as the first speaker in today's session will be to provide an overview of extreme environments, the complex whole body integrative physiological and also cognitive challenge that we see following exposure to various extreme environments. I'm all the way through going to create links from the work we do in various laboratory settings to clinical conditions that might be seen in a whole host of medical scenarios. I'm then going to set up some of the work that we're currently, we have under review on extreme environments and network physiology. So I suppose I see a lot of people in attendance today are experts in extreme environments, but maybe not everybody is. So Alireza asked me to give a brief overview of what is an extreme environment. So First thing I'd say is, if you haven't done so, the 2015 GL Brown Series lecture was provided by Professor Mike Tipton from our laboratory, very soon to be President-elect of the Physiological Society. And his topic covered extreme environments, the origins, the consequences, and the amelioration. So this is available on YouTube. It is still there. I thoroughly recommend you go and have a look. So what I've done, and I'm very thankful to Mike, over the next 3 slides, I've borrowed from this lecture because it really nicely encapsulates what is an extreme environment. If we think of our home, planet Earth, if we consider that actually 70% of this is water, the vast majority is contained within our oceans. Only 30% is actually land. And of this, if you take away deserts, thick ice, permafrost, and mountains, we're left with a rather measly 15% that is effectively habitable. That is not water, that is not desert, ice, or a mountain. And if you actually look a little bit beyond this and consider average mean air temperature just over 15 degrees Celsius, you see the hottest place and the coldest place ever recorded on Earth, ranging from -89 to over 58 degrees Celsius. We have very mountainous and very high altitude scenarios and situations such as Everest. And if we think that the mean seabed temperature is only 3 degrees Celsius, our world, our home, is actually a very extreme environment for us to live and perform in. So by its very definition, an extreme environment is a place where it is difficult to survive. And if we focus in on that word survive or survival, Mike very eloquently followed up this lecture with an experimental physiology paper also in 2015 talking about this survival hierarchy within an extreme environment. So if there's a lack of oxygen, and we'll come to that later on in today's session, whether that's by air or circulation impairments, the actual survival time is only minutes. If we consider thermal imbalance, such as hyper or hypothermia, that survival hierarchy is extended to hours. Fluid imbalance, for example, dehydration, we're talking days. And then if there's an energy imbalance, that's up to weeks. So as you can see, the extreme environments, the temperature, and the oxygen availability, we're talking less than hours to survive. In our laboratory, in the Extreme Environments Laboratory in the University of Portsmouth, we focus on the selection, preparation, and protection of people who enter into these extreme environments, whether that's for recreational purposes, occupational reasons, or even sporting reasons. And more over the last number of years, we're also beginning to consider those who are the most vulnerable in our society, whether it's the young, the older individuals, or those in our society who may have any underlying conditions. And what we focus on is these extreme environmental stressors and what happens at the whole body integrative level, whether it's things like extreme temperature, hypoxia, sleep deprivation, or exercise. More and more recently, we've been focusing on not just the physiological effects, but also the effects of these physiological stressors on someone's cognition. And a nice example, actually, this picture, the hypoxic and the high altitude example, rarely do these stressors actually appear in isolation in the real world. In reality, we often see a plethora of combined stressors, and we'll come to that later with Tom and Cecilia. The origins of some of the work that we're presenting today actually dates back to a webinar that we presented during the height of COVID in August 2020, when everything was online. And this was work or a webinar series that was hosted by Professor Tipton and myself and Dr. Matt Wills contributed on the impact of physiological responses to extreme environments and cognitive performance. And this is also still available on YouTube should you wish to go back and take a look. And as part of this, we created what we called paradigm, and we focused on this paradigm within extreme environments to really, I suppose, highlight the challenging, complex nature of when we are exposed to an extreme environment and the downstream effects of this. And we use the example of high altitude or our low fraction of inspired O2, but also this could be extended to various other clinical conditions such as hypo or hyperthermia. We know from very good MRI and PET type studies that when we see reduced oxygen availability, we will see impaired uptake of mitochondrial oxygen and non-uniform reduction of O2 across the brain. We also know that this is very likely to impair various brain networks and also the neural connectivity across multiple and varied brain regions. Undoubtedly, this is one of the mechanisms by which we see an impairment in cognitive performance. And the question we asked as part of this webinar series is, does this actually increase risk-taking behaviors? And is the physiological sequelae that occurs after exposure to various extreme environments actually sending individuals into a descending spiral where the physiology impacts the cognitive performance and the cognitive performance leads to impaired risk-taking behaviors, and it actually makes things worse? So the follow-up from that webinar series, Matt, Mike, and I published this editorial in Experimental Physiology, and we called it From Pigeonholes to Descending Spirals. And one of the take-home messages that we tried to portray through this editorial was the impact of people specializing on one very unique and I suppose minutiae of physiology. When we consider how the human body works, it's a whole host of systems integrated and working together simultaneously. So we pose the question, has the tradition of reductionist learning with increased specialization, what we term pigeonholing, meant that we've lost sight of a critical bigger picture? And through this, we've also encouraged multi and interdisciplinary teams across the broad spectrum of physiology, but not just physiology, cognitive scientists, behavioral scientists, psychologists, anybody else that has the skills necessary to answer big questions. So what we do in our laboratory has obviously various clinical links to things like hyper and hypothermia. And another is hypoxia. And this is a picture that I've made a little bit larger because obviously hypoxia in and of itself is linked to a whole host of cardiorespiratory and pulmonary conditions. But when individuals like those in this particular picture are trekking or at Everest Base Camp or even living at high altitude, it is likely that you will experience multiple and combined additional stressors, such as impairment, fatigue, most likely be exercising, likely be an alteration in the hydration status, and we may see swings of extreme temperatures. And some of the work in our lab over the years looking at hypoxia, particularly real-time SpO2 data, I noticed that it wasn't very stable or static, even in normoxic conditions. And this seemed to, from the eye, increase as the level of oxygen decreased, or the hypoxic stressor increased. So I went about looking to see if I could find any data published on this. And at the time, I could only find really one paper, and that was by our host, Mani, and his undergraduate student at the time, Amar Bogle, published in Exercise or Environmental Physiology. And this particular figure, what it displays on the x-axis is the data points over a period of 1 hour, and on the y-axis is oxygen saturation. You can see this trace is from a very healthy individual in SpO2 in the high 90s. What was interesting for me was even with the healthy individual in a normoxic environment, this particular data point or this series of data points showed some variability. And we asked the question, what happens in a hypoxic environment? Does this variability increase? So work we published in High Altitude Medicine and Biology just a few years ago now with many of the people that are presenting today looked at the impact of normal baric hypoxia at 17%, 14.5% and 12% oxygen, and compared it to a normoxic control, 21%. So we replicated the data that Amar and Ali Reza had previously demonstrated in the normoxic environment, but we extended this into the hypoxic elements. So as you can see in figure B here, this is showing mean SpO2, and as the FiO2 decreased, the hypoxic stressor increased, we see a linear reduction in individual and mean SpO2. However, interestingly, we believe for the first time, we were able to demonstrate that the standard deviation of this signal and also the sample entropy of this SpO2 signal increased as the hypoxic stressor also increased. And again, very recently, also in experimental physiology, we took this a step further and we started looking at how the various data that we were recording from a cardiopulmonary exercise breath-by-breath device, such as respiratory frequency, heart rate, SpO2, end tidal partial pressure of O2 and CO2, minute ventilation, and tidal volume. How these data were actually starting to communicate with each other using a network physiology approach, which the previous webinar went into much more detail on how this was calculated and also produced. And I guess importantly, if we extend this a little bit more, work that we recently published with the team at UCL was looking at how this extended into other conditions, such as COPD. So this is COPD data and SpO2. And what we were able to actually demonstrate was that SpO2 entropy and the fractal-like exponent have the potential to detect exacerbations in COPD. And these data here on the x-axis is the multi-scale entropy, and on the vertical, the y-axis is the sample entropy. And these data are from the same individuals, but they're offset, and you can see the stable phase in the open circles when individuals the day before demonstrated or were likely to have a hospitalization caused by aspiration. There was actually a change and a shift in how their entropy and their SpO2 data actually presented. And this is really interesting. This is now how we can start to look at how looking at our data can actually be more beneficial rather than just, I think, focusing on one narrow either outcome variable or physiological marker and pigeonholing ourselves. And this has also continued in our laboratory, where we continue to look at a whole host of different environmental stressors and how they impact both physiological and cognitive performance. And Tom will be able to take you through a journey of one particular example of what we've done in our laboratory very recently. Just a quick mention to my multiple and varied collaborators, many of who I'm sure you are aware of and know. This work wouldn't have been possible without their contribution and help. So thank you very much. And without any further ado, I'm going to stop sharing. And I think Tom is going to come straight in from here.
Thank you very much, Dr. Joe Costello. Thank you very much for that very, very interesting talk. I think I'll just introduce Dr. Tom so that we can move on with the next. So it is my pleasure to introduce Dr. Thomas Williams. Dr. Williams is a senior lecturer in the Department of Sport, Health, and Exercise Science at the University of Portsmouth, the UK. His background is in sports physiology, and he holds a PhD from the University of Portsmouth. He is also a member of the Extreme Environment Research Group at the University of Portsmouth. Dr. Williams' research focuses on cognitive performance in response to environmental stresses such as hypoxia, heat, and cold. He has expertise in experimental design in extreme environment and leads several modules in the field of sport, exercise, and environmental physiology. We're delighted to have Dr. Williams here today to share his expertise in experimental design in an extreme environment. Thank you, Dr. Tom. Please take the floor.
Hi, everyone. Thank you very much for the introduction there. And a big thank you again to Ali Reza and the guys at the Physiological Society for making this opportunity available to us today. And I'm really pleased to have the opportunity to talk to you all. Thanks, firstly, Joe, for a really nice sort of summary and overview of some of the stuff that we've done previously. What I'd like to do now, as Joe's already alluded to, is to provide an overview of a study that we've recently conducted that has attempted to put some of what Joe just spoke about there with the network physiology approach actually into practice within some extreme environment research. So initially what I'm going to start by doing is I'll talk to you really briefly sort of about how this study came about, touch on how our relationship with Ali Reza and his colleagues began to initially provide some background context. I'll talk you through how data was collected for this study that we're going to talk to you about, and then I'll hand you over to Cecilia, who's going to talk you through some of the excellent work that she's done with her analysis, and she'll talk you through our results. So I just wanted to start by saying that the study that I'm about to talk to you about and introduce now was actually conducted as part of a much larger project where we investigated the effect of combined stressors on physiological function and cognitive performance. And specifically, we were interested in examining the isolated and combined effects of acute hypoxia, sleep deprivation, in terms of total sleep deprivation and partial sleep deprivation with exercise on cognitive performance. So if that does sound like something that may also be of interest to you as well, then the details for that paper can be seen just here on the slide. And alternatively, if you fancy having a bit of a chat about it, then feel free to email me. And more than happy to always have a chat about it. So, excuse me. As Joe just previously mentioned, over the last few years, myself and Joe and some of our other colleagues have been conducting some research where we have primarily focused on investigating changes in cognitive performance that occur when we're exposed to different forms of environmental stress. And much of the work that we've done so far has primarily focused on this high altitude environment that Joe's just alluded to. And so what we generally know about this high altitude environment is that when individuals travel to the mountains, that we typically will see that they experience a reduction in their physical performance. Depending upon how high we go or how long we stay there, we're also going to see potentially a decline in their cognitive performance as well. And then what we find is that some individuals will also go on to potentially develop some sort of high altitude illness as well. And what we know basically is that the higher that we go and the quicker that we get there, then the more significant these deficits in performance are likely to be, and the greater the likelihood is that we're going to develop some sort of acute mountain sickness. And what we also know, as with pretty much all forms of stress, is that there's substantial inter-individual variability into how our body copes and then responds to this stress. So, for example, if we think of our hypoxic ventilatory response, our increase in ventilation that occurs when we are first exposed to a high altitude or hypoxic environment, we know that there's substantial variability in this response, which then has a direct consequence for many other physiological responses that are going to subsequently occur. So on the slide that you can see here, we've got some unpublished data from our lab in which across two separate studies, we exposed 21 participants to an FiO2 of 0.12 for 60 minutes. And what you can see on the left-hand side of the figure, we have our SpO2 values, which are represented by the circles. And on the right-hand side, we've got oxygenation of the prefrontal cortex measured via NIRS, or our near-infrared spectroscopy. And what we can clearly see from this figure that we've got here is that there's substantial variability between individuals despite being exposed to that identical environment. And it was this individual variability that was originally of great interest to me and Joe and some of our other colleagues. And we were intrigued to see if we could delve further to try and explain why this individual variability occurred. Could we better understand it? And it's this curiosity that eventually led to our relationship with Ali Reza and his team, where he was then able to explore our data further using the SpO2 entropy analysis that Joe just touched on, and then later on the network analysis, and then the study that we're just going to talk to you about in a few minutes' time. So as myself and Joe continue to delve into this world of looking at changes in cognition that occur in these extreme environments, in particular the high altitude environment, what became very apparent, or what we realized very quickly, is that a lot of the time when we were reading journal articles that were discussing the response to a high altitude environment, often that data came from laboratory-based studies, and that information was then being transferred to inform our understanding of exposure to these real-life high altitude environments. However, as anyone who is listening, who has taken a trip to the mountains will be very aware, whenever we travel to these mountainous environments, we're very likely to be exposed to multiple types of stressor simultaneously. And what's really important to consider when we think about that is that when multiple stressors are experienced in combination, we're then placing competing demands on multiple body systems, and therefore it cannot be assumed that the physiological response that we see in the lab where we're examining that stressor in isolation will be the same when each of those stressors are then combined and we're experiencing multiple stressors at the same time simultaneously. And so what we began to then do is to start to look at, when we travel to these high altitude environments, apart from the hypoxia, what other forms of stress were people commonly experiencing when they were in this high altitude environment? And so the first one came fairly quickly to us. It was fairly obvious. A lot of the studies, when we look at our response to these environments, are conducted while people are at rest. And again, most of us who've been to the mountains know that we actually get very few opportunities to rest, and most of the time we're performing some sort of exercise. So our first step was to begin to look at the effect of hypoxia, but when it was combined with exercise as well. Secondly, and again, those who've been to the mountains will probably be aware of this as well, is that generally when we go to the mountains, our sleep is typically very poor. And that can be due to a number of different factors. So, for example, the breathing instability that is caused by that hypoxic environment, but also often, as we can see on the slide there, it can be because we're sleeping in a tent. We might be sleeping in bunk beds in a lodge. We might have somebody snoring next to us that keeps waking us up every couple of minutes. So generally, most people who've been to the mountains for any extended periods of time will probably tell you that they got a terrible night's sleep, at least for the first 2-3 nights that they were there. So what we did was decided to design a study that would investigate the isolated and combined effects of hypoxia, sleep deprivation, and exercise on cognitive and physiological function. And at the same time as this, we also developed a second study that was going to examine the interaction between partial sleep deprivation and exercise on cognitive performance as well. So what we can see here is some of the data on the participants that took part in these two investigations. So the data from which this study that Cecilia is going to talk to you about in a second comes from two experiments that were conducted here at the Extreme Environments Laboratory at the University of Portsmouth. In study one, we had 12 healthy male participants. And then in study two, we had 12 participants once again, but we had 5 females included here as well. Unfortunately, 2 data sets were excluded from our analysis as they were incomplete, leaving a total of 22 participants that were included in our final analysis. And as you can see from that table there over on the right-hand side of the slide, all of these participants were young, healthy participants who underwent a fairly rigorous health screening to be able to take part in the study before it took place. So if I talk about what actually went on in these studies, you can see that there from study one, participants were exposed to the following physiological challenges, both at rest and then during moderate intensity exercise of 100 watts. That was hypoxia. So they were in a high altitude chamber with an FiO2 set to 0.12. They were exposed to 1 night of total sleep deprivation. And then they were also exposed to both of those stresses in combination. So both that hypoxic environment of an FiO2 of 0.12 and then that 1 night of total sleep deprivation. In the second study, measurements were again taken from the participants at rest and during moderate intensity exercise. But this time, rather than using that total night of sleep deprivation, participants underwent 3 consecutive nights in which they only had a limited sleep window under which they were able to go to sleep, so a period of 5 hours in duration. We took a whole host of different measurements throughout both of these investigations, which I won't talk to today, this is network physiology related, but again, if anyone does want to talk about the results of this study, please let me know. But in terms of the measures that are important to our network physiology analysis, we collected some cardiorespiratory data via a metabolic cart, so that included respiratory rate, tidal volume, minute ventilation, and our end tidal O2 and CO2. We measured heart rate via a 3-lead electrocardiogram, and then peripheral oxygen saturation was measured throughout using a pulse oximeter. So that's all from me for my part. Thank you again very much for listening, and thank you again to Ali Reza and the team for organising everything. I'm going to hand you over now to Cecilia, who's done a really excellent job at analysing all of this data, and she can talk you through her methodology that she used and the analysis that she's conducted. So thank you very much, everyone.
Thank you so much, Dr. Williams, for that very, very, very exciting talk. So without much ado, I'll just move ahead to introduce Cecilia. So it's my pleasure to introduce Cecilia Morandotti to our audience. Cecilia is a research assistant at the UCL Network Physiology Lab and has developed novel methods for physiological network mapping of the cardiorespiratory system in both health and disease. She's currently a student in the Integrated Medical Science program at the UCL Cancer Institute. Cecilia has a strong interest in interdisciplinary research, particularly in the application of computational physiology to medical science. We are delighted to have Cecilia with us today to discuss the non-invasive assessment of integrated cardiorespiratory network dynamics following physiological stress in human. Please take the floor, Cecilia. Thank you.
I think Cecilia is on.
All right.
Yep.
Okay. Hello, everyone. Can everyone hear me?
Absolutely loud and clear. Thank you very much,
Excellent.
Cecilia.
All right.
Excellent.
Sorry about that. So thank you very much, Tope, for the introduction. And also, I'm very grateful for being invited to talk today. So the aims of my presentation are to showcase how network mapping can be used as a non-invasive method to visualize the flow of information between different physiological variables in a system. And secondly, I'll talk about how the exposure to different physiological variables can impact system dynamics by looking at the results from the study that Tom previously mentioned. So in our study, we used transfer entropy as a measure of the directed flow of information between physiological variables. And figure A here shows how transfer entropy is calculated. So we have 2 parallel time series, each representing physiological variables, so respiratory rate and heart rate, but I'll be referring to them as A and B for simplicity. And if we want to calculate the amount of information transfer from variable A to variable B, we have to look at how at any point in time, the past of variable A impacts the future of variable B, regardless of the past of variable B. And if we then want to calculate the information transfer in the opposite direction, so from B to A, we have to look at how the past of variable B can impact the future of variable A independently of the past of variable A, again, at any point in time in the time series. So this is an example of one of the network diagrams from our study. And as Tom previously mentioned, we have the different physiological variables represented by the nodes. And the edges represent transfer entropy values. And you can see that the edges have different thicknesses, as the thickness is directly proportional to the transfer entropy value. So a thicker edge would mean that more information is being sent from one node to the other. So to sum up what I have said so far, If we want to go from a set of parallel time series, each representing a physiological variable, to a network diagram, we know that we can calculate the transfer entropy between any and all pairs of these time series, and then we can represent that information transfer in a network through a directed edge. So it's also very important to statistically validate transfer entropy values to ensure that the value represents genuine information rather than insignificant information transfer, which could be caused by random or surrogate data. So to do this, we used a Monte Carlo approach. And first, we calculate the transfer entropy that we want to statistically validate. So in this case, from A to B. And then we take the time series A and we shuffle it in order to obtain a random set of points that no longer represents authentic physiological data, and then calculate the transfer entropy from the shuffled A time series to the authentic B time series. We repeat this 100 times to obtain 100 different random time series and random transfer entropy values, which we then use to create a probability distribution. And this green line here represents the 95th percentile, which we can use to determine whether the authentic transfer entropy of A to B is significant or not. So if it is above the 95th percentile, we consider it to be significant. Whether if it is below the 95th percentile, we consider it to not be significant. And in this case, we would replace the value with 0, and it would not be represented in a network. We also use centrality measures to measure the importance of nodes, and we used in degree to measure the amount of information received by node, and out degree to measure the amount of information which is emitted by node. So this is an example of a network diagram where node size is directly proportional to the in degree value. And if we look at heart rate, we can see that it is the largest node, so the node receiving the highest amount of information. And here I have highlighted the other nodes from which it is receiving the information. Whereas this diagram is one where node size is directly proportional to the out degree value. And we can see that PET02 here has the highest out degree. And again, I have highlighted the edges through which it is emitting information to other nodes. So as Tom previously mentioned, in our study, we had different experimental interventions, and we have a total of 8 experimental conditions. They can be divided into 2 sets, one where participants were not exposed to sleep deprivation and one where they were exposed to sleep deprivation. And in each set, we have normoxia, hypoxia, exercise, and then hypoxia combined with exercise. So I will now show you the network diagrams for each of these conditions, beginning with the control conditions, so without any sleep deprivation. This is the network diagram for normoxia, and we can see that the main prominent transfer entropies are those occurring bidirectionally between end tidal CO2 and O2, as well as between minute ventilation and tidal volume. And this is reflected in the centrality measures, as these are the nodes which have the highest in degree and out degree. So if we now look at what the network looks like in hypoxia, we can see that there's a slight increase in transfer of information, and we can also see some new bidirectional transfer of information. For example, the one between heart rate and peripheral oxygen saturation. And we also see this transfer entropy from SpO2 to PETO2. And the highest transfer entropy is that from PETCO2 to PETO2. And this is reflected in the centrality measures, as end tidal oxygen pressure is the node receiving the highest amount of information in this case. So this is the network diagram for exercise. We can see that there is an increase in information transfer, and the most prominent transfer entropy is that going from SpO2 to heart rate. And we can also see prominent information transfer from PET O2 to heart rate, and bidirectionally between PET O2 and PET CO2. We can also see that there's a few new significant transfer entropies, both also bidirectionally. And we can see that heart rate here is the node with the highest input of information, as well as PET CO2, which is the node with the second highest. So if we now look at exercise combined with hypoxia, we can see that there's an increased connectivity compared to hypoxia alone, and also a redistribution in the flow of information compared to exercise. But a similarity to exercise is that the highest amount of information is still that from SpO2 to heart rate. And similarly to hypoxia, this is now a bidirectional transfer of information between the two nodes. We can also see a bidirectional transfer of information from PET CO2 to peripheral oxygen saturation, with PET CO2 to SpO2 being one of the highest transfer entropies in the network. And other prominent transfer entropies include that of end tidal oxygen pressure to SpO2. And again, this is reflected in the in degree values, as SpO2 is the node receiving the highest amount of information. So now moving on to the network diagrams for sleep deprivation. This is the one for normoxia, and it is quite similar to what we saw in normoxia without sleep deprivation, with the same prominent transfer entropies. And we can see that the highest in degree is still that between PETs, is that of PET CO2 and PET O2. This is now the network diagram for hypoxia with sleep deprivation. And interestingly, we can see that there's actually a decrease and redistribution of information transfer compared to normoxia. We can see that there's a new transfer entropy from respiratory rate to end tidal oxygen pressure, as well as a bidirectional transfer of information occurring between respiratory rate and PET CO2. And this is reflected in the in degree measure, as PET CO2 has the highest amount of information input. So if we now look at the network diagram for exercise combined with sleep deprivation, we can see that there's increased connectivity compared to the two previous diagrams in this slide. And this network is very similar to that of exercise without sleep deprivation, as we can see that the highest amount of information transfer is from SpO2 to heart rate, as well as from PET O2 to heart rate, minute ventilation to heart rate, and bidirectionally between PET O2 and PET CO2. And this is reflected in the centrality measures, as heart rate has the highest amount of information input, whereas peripheral oxygen saturation has the highest amount of information output. So this is now the last network diagram where hypoxia, exercise, and sleep deprivation were combined. And we can see that there's an increased connectivity, and there's a lot new significant transfer entropies present in the network. But the highest information transfer remains that from SpO2 to heart rate. And again, this is now a bidirectional transfer of information. We can also see significant information transfer from tidal volume to PET O2, as well as a bidirectional transfer of information between, and this is also a bidirectional transfer of information between the two nodes. And this is reflected in the centrality measures, where the node receiving the highest amount of information is SpO2, and it is closely followed by PEtO2 and tidal volume. So if we now look at the total transfer entropy values, so that would be the sum of all the transfer entropies within each network, we can see, if we look at this bar chart, we can see the total transfer entropy values represented in white for the control conditions and in blue for the sleep deprived conditions. And by looking at this, we can see that sleep deprivation caused an increase in total transfer entropy compared to control conditions. However, it did not cause an increase in hypoxia, where it actually caused a decrease in total transfer entropy. And if we look at hypoxia, we can see that it, both in control conditions and in sleep deprived conditions, it caused a slight decrease compared to normoxia. And we can also notice that exercise was the only intervention which caused a significant increase in total transfer entropy values. And we verified this using three-way ANOVA. And if we compare the total transfer entropy values of exercise and hypoxia and exercise combined, we can see that there is no significant change in total transfer entropy, but we did see that there were significant changes in network dynamics and in which nodes were receiving the highest amount of information. So now moving on to the results. So we can say that the application of information theory principles can reveal significant directional and bidirectional interactions between different components of the cardiorespiratory system. And it is a non-invasive method which only requires the recording of physiological signals. We also saw that there was increased connectivity within the cardiorespiratory networks during moderate exercise alone, or when it was combined with other stressors such as hypoxia and sleep deprivation. And during exercise, the node which was receiving the highest amount of information was heart rate, whilst peripheral oxygen saturation was the node which was emitting the highest amount of information. And this then changed when exercise was combined with hypoxia, where peripheral oxygen saturation became the node which was receiving the highest amount of information. We also saw that acute hypoxic challenge or sleep deprivation were both associated with distinctive compensatory mechanisms in the cardiorespiratory network, as we saw changes in their network dynamics. And finally, we can say that this novel network approach can be used to map the adaptive responses to physiological stressors, such as exercise, hypoxia, and sleep deprivation. And that is everything from me. Thank you very much for listening.
Thank you very much, Cecilia, for that amazing talk. I can't absolutely. Yeah, I'm blown away, absolutely. I think for now we can go to some of the questions. We've only got one question, but I've got some few questions myself. So I think this question, yeah. So, sorry. Oh, do you want to?
Can I, before we talk about question, can I just go have a very quick wrap up and mention a few things we have, and then we will ask our audience to type their question as well, right?
Absolutely.
So I want to thank everyone. So thank you, Joe, thank you, Tom, and thank you, Cecilia, for excellent presentation. I really enjoyed very much, basically. And so thank you for timekeeping. It was perfect. You know, it was very enjoyable. What I really enjoyed from this session was that, you know, it was so interesting. We had Joe, who is the head of research, associate head of research of course, a very good university, very good research center. And then we have Tom, a senior lecturer, and then we have Cecilia, a very bright undergraduate researcher who is doing well, and from medical sciences, she has learned so much computational physiology and doing great and publishing papers, etc. So that was really, really amazing. If I can summarize the whole session, which is really amazing, we learned so much from Joe talking about pigeonholing and how to avoid it. I think this is so interesting. So we are very specialized. I'm now at Royal Free Hospital in London. Basically, all of my colleagues are looking at liver, and then, of course, then if I want to talk to a physiologist, I have to go across the campus to go somewhere else. Maybe after half an hour, I can find one of my physiologist friends. So I really like this idea of, and I read your very nice editorial, and I want to encourage all of our attendees and audience to basically to also read it as well. And then the idea of hypoxia as a model, and to see how network physiology can help to understand response to hypoxia as a very important extreme challenge. That is amazing. And then Tom actually extended the work very beautifully. Tom talked about individual variability, very interesting. And also, so none of these things are really in isolation. We have everything combined, which is really, really interesting. So yes, of course, in real life, of course, we are exposed to many different things. And then how to design experiment to do that. Basically, it is I think a design experiment like that requires so much skill and also ethical consideration. And I know that in Portsmouth, of course, you have a medic of also observing, doing those very interesting experiments that you do to make sure that ethically and the safety is also observed as well. And then I really enjoyed Cecilia's very nice presentation on basically how to develop a method for network mapping based on routine data. And Cecilia, it was so interesting to see all of those beautiful pictures because you made difficult concept easy for us. And I liked the presentation, and probably I will borrow your slides for my future lessons, etc. So I'm really grateful. And so I have lots of questions myself we can ask, but we have questions. So Tom, so Tope, would you like to go through the question? Is it okay?
Yeah, no, absolutely. So thank you very much for that wrap up, really. So the first question we've got in the chat, I think we've only got one, but of course, some few questions that'll be coming later. So the question is coming from Shantila. I hope I pronounce your name appropriately. Apologies if I don't. Did you observe? So the question is, did you observe any significance inter-individual variability in the network pattern? I'm assuming that this question is directed to Cecilia or Dr. Mann, if you want to take that question.
So I think there are definitely some changes, some differences in the network dynamics between each individual. But in our study, we ended up using the median of all of the network dynamics and centrality measures in order to look at the broader image rather than in individual patterns.
Absolutely. Thank you very much. Does anyone want to add more to polish that? Thank you very much, Cecilia.
I just want to mention something that actually, so Cecilia is collaborating with a very good group in Pescara, Dr. Danilo Bondi. And so I think they are studying that very well. So the idea is, what about the variability within healthy people at this time? And I know that they have excellent, very interesting results. And of course, they haven't published it, but they will publish it soon. But so I know that someone is trying to, that group are trying to look at it, and I have seen some of the results and are really interesting. Yes, it is a very good question.
Thank you very much for clarifying that. I think the next question has come through from Matt. So Matt is asking, thank you for a fantastic webinar. Thank you, Matt. Broader question. Has this approach ever been applied to patients under anesthesia, where some of the physiological system can be directly controlled? So anyone want to approach that question?
I have some comment if possible.
Yes, please. Thank you very much.
So Matt, thank you very much. Thank you for attending the webinar as well. So basically, so yes and no. So I think we have done a study recently published. Actually, Cecilia is the first author of the paper published recently in Journal of Applied Physiology about patients with sepsis in the intensive care unit. But some of those patients, they have mechanical ventilation, of course, like many other patients in the intensive care unit. And then this very similar network mapping technique has been used. We can actually maybe share with you the paper later. But then, yes, if we compare patients who have mechanical ventilation, the transfer entropies are reduced significantly. And so, of course, then when the body is not controlling it, when we don't have intrinsic mechanism and we have extrinsic mechanism such as mechanical ventilation, of course, you can see a significant drop in those transfer entropy and network maps. This is already there, but it is not necessarily in patients with under anesthesia. I guess the anesthesiologists have lots of data because, of course, the monitors are there, but I'm not aware if anyone has published that. Can I ask Cecilia, have you seen any paper on patient with under anesthesia with network mapping?
No, I haven't seen any, but that is a very interesting like view to look at it from.
Absolutely. And if I may just chip in as well, I think, like you rightly said, Ali Reza, the idea of the sepsis patient, and I'm assuming that the data is coming from Mimic. If the data is coming from Mimic, for instance, I'm assuming that there will be data available, or there'll be variables available where you can see which patient. So it's something that can be looked into, absolutely. But it's just an amazing question to just to wrap up, and it's something that we'll probably would like to see or look to do in future.
Thank you both very much. I guess the follow-up to that would be, I was thinking more about if you did it in something like day case surgery, where the people are actually well, but might be ventilated or not, and then you could, in a large data set, and you could potentially do this prospectively, so predict what you're going to see, and then follow up from there. But that was a brilliant webinar, and Cecilia, you explained something very complicated in a way that I could understand, and I was incredibly impressed. Thank you.
Thank you so much.
Thank you. I think Joe has Joe has a comment, please.
Yeah, thanks, Matt. Great question, and I think there's a funding application in there for somebody, whoever wants to put it together. I think that's really important what you said about the example about the day case, because what you effectively have is you've got a healthy control condition where you can directly compare, and somebody else asked about the intra-individual or inter-individual variability. And a lot of what we have, the data that we have from laboratory setting, we effectively create patients from healthy individuals by delivering an extreme stressor, which allows us to compare back to a control setting as well. So your example there of day patients is a really nice one, and it might extend this work beyond the lab and into a more clinical setting. Thanks.
Thank you, Joe. Thank you very much. So I remember when I was we were doing some literature search for SpO2 variability, and then we came up a very interesting report going back to 1990s. Of course, they didn't use entropy analysis, but it's a report from an anesthesiology group at Cambridge, one of the big hospitals in Cambridge. And what they observed that actually, so after general surgery, then there is instability in SpO2. And then they try to measure it using different ways. They use, maybe, of course, a Fourier transform, etc. But that was a report without knowing actually what's happening. But then it goes along very well with all of the data that actually Joe showed us about when we have hypoxia, entropy goes up, etc. Maybe there is hypoxia or there's a problem with the network after the surgery. I know about this report. It is not exactly network mapping, but it is a very nice approach from a rather old paper now. And we usually cite this paper in our publication because it is the first time that that kind of instability was reported. They use the terminology instability rather than entropy. Thank you. Tope, I think you are on mute.
Yeah, sorry, absolutely. So there's one question from Ella. So Ella is asking, thank you all three for a very interesting presentation. So Cecilia, this is directed to you. So what software did you use to run your analysis? I think that's the question to Cecilia.
Okay, so for the transfer entropy calculations and creating the network diagrams, we used MATLAB. And then for the statistical analysis, we used SPSS.
Thank you very much. I hope that answers it, Ella. I think there's another question as well from Yao Peng. So the question goes thus: thank you very much for your insightful presentation. I have a question regarding the calculation of transfer entropy. When computing transfer entropy, it is necessary to set time lag. In your study, is the time lag fixed for all links, or is it selected differently for different connections? Also, does this time lag have any physiological meaning? Thank you very much, Yao Peng. I think you've covered one of my questions, actually. So, yes.
So the question for Cecilia.
Okay, yes. So that's a very good question. We used the same time lag in all of our transfer entropy calculations, and we specifically used a time lag of 5, and the way we decided that was by trying a set of different time lags until we found that there was a plateau in the transfer entropy values which were calculated with those different time lags, if that makes sense, I hope.
Absolutely. I can rub off a little bit on that as well. I think the idea is you choose different time lags, probably from 1 second to maybe 15 seconds, and then you calculate the transfer entropy for individual for each one of those time lag, and then you plot it. So now you're going to have on the x-axis the time, and then on the y-axis you have the transfer entropy magnitude. So then if you then plot it, you look at where it peaks, and then once it plateaus, you know what value gives you the highest transfer entropy, and that's what you go for that specific. I think I may have covered it a little bit, but maybe Alireza, you want to come in and add a little bit more
I
information.
agree 100% with Cecilia and on you, of course. That's thank you for the very good technical question. Very good.
I think that's the end of the question from the audience.
Do we have time for? I have some questions. Because we have this excellent panel, and I want to ask maybe all of our speakers a question about what Joe mentioned about pigeonholing and how we are so specialized. How can we solve this problem? I want to ask your views, if possible. So, for instance, what do you do in your because for instance, I know that Joe successfully collaborates with very good people. I know he has collaborator in Japan. I have seen many very interesting papers and works, cross-disciplinary work. So, and it has. I want to know your view, Joe and Tom and Cecilia. How, if right now that we have so much super specialization, how can we solve it to have good discoveries? So Joe, can I ask your opinion?
Yeah, thanks, Alireza. It's a really good question, and I think there's a number of ways of looking at this, and a lot of science and research comes down to two things: time and money. So if we think of money as a first starting point, and we look to different funding bodies and our pillars of funding, certainly in the UK system, I'm conscious that we've other people from outside the UK here as well. Many of the, whether it's ESRC or EPSRC, or whether it's the MRC or various different key funders like that, are very effectively themselves pigeonholed in what they fund and the areas they fund. Very recently, UKRI held a cross-council research bid, our funding call, and they were inundated with the volume of applications that went in. So there's a lot of interest in doing multi and interdisciplinary research, but it's often a challenge. What I would say is, and this is quite a nice example, and I think Alireza, you spoke to it very nicely in terms of the breadth of people, from professors to undergraduate students that have worked on the various collaborative projects that we've done. But I think fundamentally, it's just people who have an idea and have an interest, and they're aware of something they would like to do, but may not have the skill sets to do it. So we're very much human and applied physiologists that work in an extreme environments laboratory. and our collaborative networks, excuse the pun, our collaboration basically came from reaching across the divide, even within physiology, in the area of computational physiology, and saying we've got all these really interesting data and ideas, but you've done the work from a computational perspective, and actually encouraging people to collaborate. So if there are any ECRs or early career investigators or undergraduate, masters, or PhD students, sometimes think outside of your field, think outside of your direct field, and actually think how it might actually contribute to other areas, be it in physiology or even wider.
Thank you. Such a good, interesting idea. I like the idea that you also talk about science policy and funding as well, because of course sometimes a discipline is really pushed because of funding is there, and sometimes a discipline is not really progressing because there is not much funding, but although it's very interesting and maybe in future it has lots of. Thank you. That's a very interesting and very interesting comment. Can I ask maybe Tom or Cecilia? So what do you think about it? Because so from Tom, because I know that Tom, although you work on extreme environment and in a sport, maybe physiology, and you have a sports physiology background, but you do lots of neuroscience work, right? So then how could you come up with solving the problem of doing a neuroscience project within the sport? Can I ask your opinion? And how can you facilitate this kind of collaboration?
Well, I don't really know how useful this information is to anyone, as it seems fairly basic. But Joe's advice to me throughout my PhD was effectively, if you don't know how to do something, find somebody who does know how to do it and strike up a conversation with them and have a chat and find out if they want to be involved with whatever it is that's going on. And again, that sounds fairly simple, but it does always surprise me how many people then continue to try to do something, sometimes not very well, without then just simply reaching out and asking other people who've got lots of experience in that area to help them out. Exactly as Joe just said with those data sets, where we sort of had conducted a few experiments, had lots of data, a few ideas, but no idea how to do them. You had all the sort of know-how and ways to sort of process that data and look at it, and just a simple conversation from you and Joe has now basically sparked several papers together, this webinar, and yeah, it's just amazing what will come from just a simple conversation, I think.
So the conversation is always.
Don't know how helpful that is.
Okay.
Talk to people.
Thank you.
Sorry,
Aliras, we've got some extra questions coming through the chat.
But after after question, I want to know Cecilia's view as well. But of course, questions, please.
Yeah, no, absolutely. Thank you very much. So, Thad is asking that, following up on Matt's question, it seems that uncoupling of signals utilizing agonist and antagonist could also be of benefit in interpretations. That's a comment from Thad. Anyone wants to tackle that?
I like this question. As a pharmacologist, I really like it, because of course what Matt mentioned is about, okay, so sometimes in anesthesiology we use mechanical ventilation, etc., but also we uncouple organs by giving beta blockers, alpha blockers. We use a lot of agonists, antagonists, and this is interesting. So basically, of course, a very good example of it is that actually whenever we anesthetize an animal, for instance, for lots of assessment in physiology, most of those variables are gone. Heart variability drops to almost nothing, etc. I think it's a very good and fair point. But I'm not aware that if pharmacological manipulation has been studied. But I assume that maybe we have, of course, the world expert in network physiology, Professor Ivanov, probably with us in the session. And I know that, of course, he has much better overview of the field, and in case there is any study, of course, probably he knows better than me. But I think I say that it's a very good idea. Of course, that's the same idea. You can uncouple by pharmacological manipulation as well. And some of the studies that they use reductionistic approach, basically they have to anesthetize, they have to, of course, maybe block the neuromuscular junction, and then we don't really see the full network because everything has been isolated already. And I know that studies on especially cognitive, of course, neuroscience studies, they suffer a lot from that kind of pharmacological manipulation, and what doing something without any pharmacological manipulation sometimes is very difficult. Thank you for that type of discussion.
Thank you very much. So I think there's other questions as well. So this question from Lujin. Thank you for the very interesting talk. I would be keen to read a short summary of the presentation for wider dissemination for this series. I think that would probably be directed to the organizers, the Physiological Society, in terms of probably providing a summary. I'm sure that this video will be available on YouTube for future references as well. But the idea that Lujin is putting forward is potential for probably a summary of the presentations for wider dissemination. I think it's something that we can work on. Thank you very much, Lujin, for that very good suggestion. I'll move on to Tariq. So Tariq is asking, thank you very much for these very interesting presentations and topics. Is there any interest in applying these signal transfer findings between the different organ systems and microorganisms, billions which are living in the human body? So that's the question from Tariq. So yeah.
Would you like to answer? Do you have a because we can that is actually absolutely interesting question. You know, the gut-brain axis, gut microbiota, all of those things. So then, what kind of signal we receive? So of course, there are lots of things to think about it. Really, that's a very good question, probably for future. But so combining metabolomics, because of course the bacteria in our gut, for instance, of course, they make lots of metabolites. Combining metabolomics with, of course, with network mapping would be very interesting. Of course, I think this is a very, very good question.
Yeah, no, absolutely. I do. Yeah, I do agree with you. I think potentially the approach will be just to summarize, Tariq. I don't think there's any research in network physiology linking organ system interact with specifically with the potentially the microbiota in the body or in the system. But like Ali, my colleague said, it's an absolutely amazing approach just to see probably the difference in potentially the network connectivity between those with a certain level of microbiota, or to look at is there any links between how much microorganisms you have in your system and the healthiness or the strength of the connectivity between your organ system. I think it's something that should be looked at. I don't think it's been looked at, but thank you very much for that specific question.
So can I just take the opportunity to advertise for our future, basically. So we are talking about sepsis, of course, interaction of bacteria and physical system. On next week, Emily Ito, one of our excellent lab members, is going to talk about it. And also when we have a talk about cirrhosis, of course, microbiota plays a very important role in liver cirrhosis, in liver failure. Next week, we have another session, which maybe we touch a little bit about this topic as well. And I'm very happy that Joe, would you like to maybe say something about this question, please?
Yeah, it's kind of a follow-up to the microorganisms, but it's kind of at the other end of the spectrum. Again, I'm going back to the whole body integrated physiology approach, and it seems like a basic thing I'm going to say, but I think it's just more evidence, and it's 101 exercise physiology, and I'm conscious that there's a lot of exercise physiologists on this call. But these data, again, when we look at extreme environments, whether it's full overnight sleep deprivation or partial sleep deprivation across multiple nights, or relatively moderate to high levels of hypoxic exposure, exercise always comes out as a very potent stressor. And I think this is something that we should never lose sight of, that exercise is in of itself a very potent stressor. And stress is not always a negative thing. In this instance, it's a very good example of how humans, and I know our colleague Mike Tipton has wrote on this many times, and he's a very nice editorial in experimental physiology, saying that today in society and technology and our intellect have created a scenario and environment where humans, their is very homeostatic. There's very rare, I suppose, need for an adaptive response, whether it's temperature or whether it's exercise with technology and vehicles, etc. So when we are looking at challenges to an individual, I think we need to look at stress not only as a negative thing, but how it can be positive for adaptive as well. And I think there's potentially a lot for the growth of network physiology in those spaces too.
Thank you. That's amazing. Thank you very much, Shady.
Thank you. I have one last question. I think it's for Joe, Tom, and Cecilia. Have you looked at the potential for integrating other time series such as EEG and EMG into your analysis in future, especially because we're talking, I think in the beginning of the talk, there was a lot of focus on cognitive changes in extreme environment. Is there a plan for in the future to look at things like EEG, especially in the simulated environment in your labs, and how they change across all those stress or exposure?
I'm happy to go first, and then perhaps Tom can jump in, or Cecilia. We certainly have had multiple conversations, just to pick up on your EMG, with our biomechanists as well, looking at different stressors and extrapolating some of this data and these ideas. One thing that I guess is very, I suppose, to contextualize a lot of our data, we're also limited by the outcomes we collect. And Cecilia talked at length about SpO2 being kind of the centrality node, and a lot of information, the bidirectional playing to that. I do think it's very. I think physiologically that SpO2 is the central node and the most important thing. I think we're using it as a surrogate for other higher, kind of more central areas of oxygenation and central. So if we had that data, the challenge is getting it synced and in real time. Things like EMG, you can do that. ECG, absolutely. But the breath-by-breath CPET device that we utilize, there's only so much kind of non-invasively that you can get. So I think really to step this on further in terms of extreme environments, we need to think about how we can measure things in real time and sync them. That will be the challenge. That will come with technology again.
Thank you very much.
Thank you very much. Thank you. Tom, do you want to add anything?
No, the only thing I potentially maybe to have is we do have some nice data collected using an EEG. That was actually part of this study that we just discussed there. So whilst we've collected the data, I'm very aware of putting that load then onto Ali Reza and Cecilia, saying they can perform the analysis. But yeah, we do have some more data potentially that could expand this beyond what it is now, and start maybe looking at some other measurements as well. Yeah.
Thank you. I mean, thank you very much. Thank you.
Yes. Sorry, if you don't mind, I'll quickly read some. So the other comments. Sorry, apologies, on the chat. So Ella goes again. Says regarding avoiding pigeonholes, I've been fortunate to work with Joe and Mike T. My observation was that not only are they incredibly personable, they have a wonderful scientific curiosity. I think this makes them very successful in working with those from various scientific backgrounds. And it's another comment. Thank you very much, Ella.
100% agree. Thank you.
Yeah,
Thank
absolutely.
You, 100%.
Yeah, Professor Ivanov also have a comment in the comment. So comment related to previous questions. His comment goes, does network physiology transcends scales and levels of integration from subcellular and cellular signaling and interactions to metabolic and proteomic networks to organ system interaction to understand functions and state at organism level. So the idea is, yes, there's potential to be able to look at the network connectivity across different scale. Of course, the idea would always be, and the limitation, like Joe rightly mentioned, is the technology. Do we have enough technology at hand to be able to translate those into our research lab, essentially? But thank you.
Thank you, Prof. Ivanov. Thank you. Thank you very much. Thank you very much. Okay, so I think probably I need to show you a slide about next week's talks. And basically, after that, of course, we can conclude. But I think, is there anything remaining? Because I think Cecilia, we didn't ask Cecilia's comment about integration. And in case, Cecilia, do you have any comment about, because you are, of course, younger than, of course, very, very bright, but basically, of course, is there any, so how do you see integration? Because maybe you registered for a very specific course in university, but knowing where from other discipline is important. And for instance, you have done lots of computational physiology. Do you have any comment about how we can actually bridge things to make things more integrative?
Yep, I think one of the important things is like for undergraduate students like me, just allowing students to not like focus only on the like specific topic of their course. For example, I initially applied to a cancer biomedicine bachelor's degree, but then I saw the work that you were doing and I found it interesting. And again, just like maybe connecting with different professors or lecturers that are experts in other like other fields that interest you as a student, and just like maybe widening your knowledge.
Absolutely. Thank you very much. If I may just quickly add something, I think it might be the opportunity to sort of mention one of the UCL approach. I think one of the reasons potentially why UCL is so successful in being able to bring along, especially the undergraduate student, is I think there was a book by Dilly Fong. It was Connected Curriculum. I think I'll invite some of our audience to go and have a look at the book. So it sort of looks at the approach to teaching in such a way that you can bring along everyone. And the idea is that the research that is going on in every faculty, students, especially learning students, should be exposed to the highest level of whatever is going on in the department. And that allows them to look at the length and breadth of what is actually being taught. And then, of course, to consider some of the opportunity if they, and it sort of stimulates some of their interest as well going forward. And then, of course, widening participation, like Cecilia mentioned, to be able to allow those students to get on board and contribute. It sort of creates a sort of incubator, essentially, for the future, because obviously we're all doing amazing work, but you need people, you need continuity. And being able to bring along the younger ones, I think, is very crucial, essentially. Sorry.
Thank you very much. Okay, so maybe I may share my slides just to apologize. Where is the slide? Okay, this is slide. Okay, basically, so okay, so thank you all, and for excellent comment, and also thank you, Professor Ivanov, for mentioning about the of course dimensions of network physiology. We have lots of things to do for future. Amazing. We have lots of methods to develop, lots of things to study, and of course the discussion after our wonderful talks today show that lots of things needs to be done, and I'm happy that we have this opportunity to discuss it thanks to the Physiological Society. So today, this was the talk that was highlighted network physiology in extreme environment. If you're interested in that kind of discussion, we have two excellent sessions in next week, 2nd of April, Wednesday, and 3rd of April on, of course, Thursday, exactly next week. So we will have next week on Wednesday, we have a network physiology in respiratory disease and in critical care. So we will have two talks, one by Dr. Amar Shah, a consultant in respiratory medicine, talking about sleep apnea. And he has done a very interesting work in sleep apnea and also application for network physiology. And what Joe mentioned about COPD, of course, it's very relevant to this one. Of course, we are talking about another common respiratory disease that involves lots of different systems. And then we have Emily Ito, one of our excellent graduates who has done lots of interesting work on sepsis, and she's going to talk about sepsis, and I'm sure you will enjoy it. So if you haven't registered, we have, I think the registration closes on Monday, 31st. So please register to make sure that you don't miss the opportunity to come to the meeting and discuss and ask your questions. So, and also then our last webinar of this series is on Thursday next week, 3rd of April, and we have invited Sara Montagnese, a chair of medicine and hepatology at the University of Padua, a very, very good clinician and also excellent scientist, to talk about organ involvement in liver cirrhosis. And also, of course, my co-host, Tope, is going to talk about application network physiology in liver cirrhosis. So please register if you haven't, because the registration is still open till Monday, if I am not mistaken. There is just one technical thing. So because this weekend, I think we are going to have a daylight saving time, so we are going to take 1 hour forward in the UK. So we are going to do the webinar next week at 3 p.m., not GMT, but British summertime. So if you are in the UK, that's fine. Of course, we are following the same watch. If you are joining us from other countries, please check your local time with this time. So next week, we are going to join the session 3 p.m., the British summertime. Please make sure that the local time is okay. And I think we have only just maybe a few minutes. I want to thank especially Abby, Simon, Caitlin, and Sarah from the Physiological Society, because they have been so wonderful to actually make this event possible. So for us, we just focus on the science, and then they did all of the work with registration and making the platform ready for all of us.
Absolutely.
So I don't need to share anything. Is there any final word?
Yeah, no, absolutely. I just want to quickly mention, not to forget that there's another physiological society, a seminar that is coming on, the one that is organized by Professor Ivanov as well. So it's just I think a lot more information is available online as well. So for those who are interested,
Exactly.
I'll leave a link here as well for those who are interested.
Yes, there is a blog about network physiology. And of course, if you just type network physiology webinar, the blog comes, and then the information is there.
Absolutely, and I'll leave the link also in the chat for anyone interested. Thank you.
Okay, thank you very much. Perfect then. So again, thank you very much. Thank you, Joe, Tom, and Cecilia, for excellent discussion and this excellent talk. And thank you all of the attendees for coming and asking questions. So we hope to see you next week. Have a wonderful weekend, and see you next Wednesday. All the best.
Thank you. Thank you.